Hello and welcome to this Integrated DNA Technologies webinar, Unveiling the Potential of CRISPR, a guide to gene editing. My name is Yoonhee Jang, and I'll be serving as your moderator for today's presentation. The presentation will be given by Edward Wang. Dr. Edward Wang completed his PhD at the University of Adelaide. He worked as a postdoctoral fellow at the National Cancer Center Singapore for 3 years and published the findings on the predictive factors of genetic screening of BRCA1 and BRCA2. He also published a study on the importance of genetic testing for 25 breast predisposition genes using NGS. The presentation should last about 30 minutes. Upon completion, I will lead a question and answer session where the speaker will address as many questions from the attendees as possible. As attendees, you have been muted, but we encourage you to ask questions or make comments at any time during or after the presentation by entering your questions in the Q&A box at the bottom of the screen. Also, please note that you can expand the presentation slide window for easier viewing. In case you need to leave early or want to revisit this webinar, we are recording the presentation and we'll make a link to the recording available on our website a few days after the presentation. You will receive a link to this in our follow emails.
Thank you, Yunhee, for the introduction. Good day, everyone. My name is Edward from the IDT. In today's presentation, I will be sharing with you the CRISPR technology and its relevant products that IDT is offering. Let me walk you through the contents of my presentation today. I will give a brief introduction about the CRISPR technology, followed by the introduction of CRISPR solution that IDT is offering, known as the altered CRISPR. And lastly, I will be wrapping up my presentation today with the applications that CRISPR has been adopted to date. Now, let's get started with the introductions of CRISPR technology by taking the Cas9 as example. CRISPR is an acronym of the clustered regularly interspaced short palindromic repeats that was discovered in the early 2000s. Functioning as a defense system in bacteria against the foreign materials such as pathogens nucleic acid. Over the years, scientists managed to discover and identify the essential components in the system for genome editing purpose. That includes the programmable RNA guided endonuclease known as the Cas endonuclease that targets the DNA in a sequence-specific manner and a guide RNA sequence. The Cas9, as shown in the shape of blue sphere, is the flagship CRISPR system and still the most commonly used CRISPR enzyme. The guide RNA of Cas9 system consists of a cirRNA and a tracer RNA or scaffold. This cirRNA consists of a 20 to 24 nucleotide spacer shown in the light blue line to determine the specificity, while the tracer RNA helps to mediate the binding of Cas9 to the region of interest. The spacer sequence is adjacent to the PAM sequence, known as the protospacer adjacent motif. The PAM sequence can be varied depending on the Cas endonuclease used, and the commonly known PAM sequence for SP-Cas9 is NGG. After the formation of CRISPR-Cas complex, it can then trigger its cutting ability to form a double-strand break. This double-strand break can be repaired by either the non-homologous end joining, or known as the NHEJ pathway, or the homology-directed repair, or known as the HDR pathway. In brief, when there is no donor DNA template, the double-strand break will be repaired by the NHEJ pathway, of which the 6 random nucleotides around the break will be deleted that cause the indel, resulting in a frame shift and subsequently leading to the knockout of that gene. On the other hand, when there is a donor, either the use of a short donor oligo or large gene fragment that flanks with the homology arms at both the 5' and 3' end of the donor. What the cell will do is to use this donor as a template because the homology arms are already there, and the region in the middle will be repaired into the break. And through this approach, it allows the researchers to introduce the point mutations, to tag the gene, or even to correct the genes. IDT offers a complete workflow begin with the experimental design along the way to the analysis part at the end. These products include gRNA and donor template design tools for the Cas9 system, wide ranges of the CRISPR-Cas reagents including the custom gRNAs for the Cas9 and Cas12a nucleases, the altered nucleases themselves, as well as the frozenly labeled Cas9 tracer RNA and the electroporation enhancer, different types of the donor template, and lastly, the toolkits to analyze the genome editing efficiencies, which I will be discussing in the later part of the talk. As mentioned earlier, to achieve the genome editing through the CRISPR-Cas9 requires the introductions of the ribonucleoproteins, or known as the RNP, into the nucleus of a cell. As illustrated here, there are many ways to introduce the CRISPR-Cas9 components into the cells, from using a combined expressions plasmid to co-delivery of a Cas9 mRNA and gRNA, using the Cas9 expressing cell lines and transfecting with the gRNA, and lastly, the pre-assembled RNP complex with a purified Cas9 proteins and the synthetic guides. How the reagents are delivered, the format of delivery matters, and we recommend delivery of in vitro assembled active RNP complexes with the advantages of lower off-target editing rates and DNA-free to simplify and accelerate the entire genome editing workflow. Currently, there are two guide RNA formats to consider when implementing the CRISPR-Cas9, the two-part guide RNA and the single-strand format of the guide RNA, or known as the sgRNA. On the left, the two-part guide RNA, the cRNA contains the protospices, or known as the guide RNA, sequence and determines the specificity, and the tracer RNA is a universal sequence that required for Cas9 binding to the guide RNA. These two pieces need to be annealed to form the guide RNA. Another option for the guide RNA is the single guide format, or the sgRNA that I mentioned earlier, which is a one-piece RNA oligo at 100 ml in total. This is a great choice for any application, especially for challenging conditions, and it adds the convenience of not requiring the annealing step. All guide RNA options available at IDT are chemically modified for increased stability and to reduce the risk of triggering the native immune response. IDT developed different guide RNA formats for various needs that are suitable for the Cas9 and Cas12a systems. For Cas9 system, we have the altered two-part standard, the altered two-part XT, and the altered sgRNA. The altered two-part standard refers to the cRNA that consists of standard modifications that require to stabilize the guide RNA in the cell and suitable for most of the cell types. The cRNA XT is chemically modified for additional stability, whereby the sgRNA, as mentioned, is a 100 ml long guide RNA with modifications that require for the stability. Both of these two different types are particularly suitable for the applications of A, to co-delivery with the Cas9 or the plasmid or the Cas9 mRNA, and B, the RNP under difficult experimental conditions. The guide RNA of Cas12a is about 40 to 44 nucleotides in the total length. At IDT, our guide RNA is optimized that includes the conserved regions that suits for different types of the Cas12a that purified from different strands. In a nutshell, IDT offers different versions of the guide RNAs, allowing the researchers to better meet the needs of their applications and control the costs. There are many design tool options available, and I would encourage you to take a look at the IDT Guide RNA Design Tool, which is very user-friendly, flexible, and provides a platform for single or batch designs. The design tool has a library of pre-designed options for various species, including the human, mouse, rat, zebrafish, and C. elegans, and the tool can accommodate custom sequence requests. Here is a screenshot of the design tool preloaded with a design search for the MYF5 gene in zebrafish. The input can be gene symbol or accession number, and the tool will generate the exon-intron map of the genes, with the exons in a thick bold in color and the introns in a thin lines. The guide RNAs are indicated by the arrows, along with their direction. The tool provides a ranking list of guides along with a guide RNA on or off target score, which can range from 0 to 100. 100 being the best score for both on and off target. After the generations of the guide RNA design, the next step is to choose your guide RNA and the Cas9 reagents. This is the screenshot of the previous inquiry. You can see the guide RNA sequence, genomic positions, and the on or off target score. And you can expand the off target details to take a look at the potential off target size. The tool also integrated the option to select the Cas9 nuclease, and at IDT, we have different options of Cas endonuclease to choose from. In addition to Cas9 and Cas12a guide RNAs, IDT can support custom CRISPR guide RNAs for any alternatives application or the system, including the prime editing, Cas13a, and the others. With the custom guide RNA design tool, we have enabled ordering of custom guide RNAs that up to 150 mR with the ability to customize the full sequence to fit any novel CRISPR nuclease. Also, it provides customers with the flexibility to select the quantity and modifications that are required for the experiments. This is the data that we conducted internally, demonstrated the advantages of using the RNP approach than plasmid for the genome editing. As you can see from the data here, plasmid-based genome editing can last more than 72 hours, resulting then the increase of off-target rate. On the other hand, the RNP complex will degrade after 24 hours post-incubation. Therefore, lesser off-target rates and toxicity, more controlled and simplified the entire genome editing workflow. In this study, we compared the editing efficiency between the plasmid-based Cas9 and the protein-based Cas9 that complex with the two different guide RNAs. The orange color indicated that the on-target editing rates, while the blue color indicated the off-target rate. The data on the left panel show that with plasmid-based approach, high off-target efficiency was observed. On the other hand, the RNP approach can overcome this limitation to increase the on-target rate while reducing the off-target rate. Another highlight shall be the improved versions of the high-fidelity Cas9, which is a variant of wild-type that developed by IDT. In this article that we published in Nature Medicine, it demonstrated the comparison study between the wild-type Cas9 and the high-fidelity Cas9. The author described the off-target editing, such as translocations, when using the wild-type Cas9. But the high-fidelity Cas9 that IDT is offering, the data shows that it helps to reduce the off-target rate while maintaining the editing efficiency. Now, I would like to discuss about the delivery approaches for the RNP system. Generally, the RNP can be delivered through different approaches: the liposuctions, electroporations, and microinjections. Each of these delivery methods possesses its own advantages and disadvantages, which I will discuss in details. Liposuction is commonly used among the researchers for transfection. This approach is convenient that requires to mix the RNP with lipid transfection reagents such as the CRISPR-Max or RNAi-Max, followed by incubating with the cell in culture. This approach is usually cheap and doesn't need expensive equipment. Additionally, it uses a small quantity of CRISPR RNP. Generally, 2 nanomole of gRNA is sufficient for more than 1,000 reactions in a 96-well format. The drawback of this approach is it not feasible in all cell types, especially sensitive cells such as the primary cells or the iPSC cells. The electroporation approach is most reproducible method for cell culture work and generally robust and consistent for difficult cell lines. It requires the mixing of RNP with the cells in suspension and electroporation buffer. An electric current is applied to perforate the cell membranes, allowing the delivery of RNP into the cell. The drawback with this approach is higher cost that requires expensive equipment and consumables, and it uses more RNP than liposuctions. Generally, 2 nanomole of gRNA is sufficient for about 15 reactions using the 4D nuclear factors from the Lonza. Other methods are used for different applications, including the microinjections for in vivo delivery into the embryos, the ballistic bombardment in which the RNP are coated onto the particles and shot through the cell walls, lipid nanoparticles in which RNP are encapsulated in lipids for cell culture or in vivo delivery, and CRISPR Gold, which the RNP are bound to the DNA conjugated gold particles, then coated in a polymer to facilitate the delivery into the cells. At IDT, we have a library of protocols available on our website and the references to help you get started. These protocols were either developed internally from our IDT's R&D group or submitted by various other users. In addition to these protocols, we have a deep support team to help you with the genome editing with CRISPR. In addition to internally developed and users-provided protocols, IDT has continued to improve the innovate novel reagents to further improve the efficiency of delivery. One classic product is the electroporation enhancer. Here's a quick look at the Cas9 electroporation enhancer and how it improves the delivery and editing efficiency. The Cas9 electroporation enhancer is a single-stranded DNA oligo, or known as a carrier, that you add to the RNP mixture before the electroporations. We find that the additions of this carrier DNA can significantly boost the editing efficiency, particularly when using the suboptimal dose of the RNP or at challenging sites. On the right is an example of how this behaves at one site in the two-cut cells. The blue line represents the RNP delivery without the electroporation enhancer and shows a linear increase in editing efficiency with an increased dose of RNP up to 4 micromolar. In contrast, when using the electroporation enhancer, we can achieve the maximum editing efficiency at a much lower dose, in this case at 1 micromolar of RNP. Using a lower effective dose can reduce the off-target events and also lowers the reagent cost. The use of the electroporation enhancer is something that we highly recommend in all experiments. Delivery is the most common bottleneck in the CRISPR experiment, and having the ability to visualize and track the RNPs can be a great way to check that they were successfully transfected. IDT offers fluorescently labeled Cas9 proteins, which can be used for enrichment of successfully transfected cells. Additionally, IDT offers the fluorophore-labeled tracer RNA. This can be used to visualize the RNPs under the microscope, as shown on the upper right panel, or used for cell sorting applications, either for enrichment of the transfected cells or for optimizing delivery. Next, I am going to provide a brief overview about the array-based gRNA libraries that offered by IDT for screening purpose. For CRISPR screening using the array's cirRNAs, these individual cirRNA sequences targeting different genes are kept separately in different wells of a multi-well plate. These RNAs are then combined with a Cas enzymes to form the RNP complexes, followed by the electroporations into the cells in individual wells. CRISPR genome editing knocks out different genes of the cells in different wells, and the edited cells will then be treated with either the drugs or other stress conditions for positive or negative selection purposes. Observations of the changes in a known phenotype or response in different wells of the cells is used to determine which genes are involved in the physiological effect being studied. IDT supports the CRISPR libraries with different gRNA options, including the pre-designed and custom design options, and a custom formulation services. CRISPR libraries can be two-part or sgRNA, put by the genes in a multiple gRNAs in a single well, or fully arrayed, which means one gRNA per well, and can be provided in either the 96 or 384 well plates. Next, I would like to introduce our HDR reagents for generating the CRISPR knock-ins. IDTs offer different formats of the HDR reagents that can be used in different scenarios, including for small edits, we have the modified HDR donor oligos up to 200 mers. For large insertions, we have the megamer, which is a long single-strand DNA, and now we expanded to a modified double-strand DNA donor, known as the altered HDR donor blocks product, allowing for sequences up to 3,000 base pair and of greater complexity. And lastly, IDT has also established the HDR Enhancer V2 to help improving the knock-in efficiency. At IDT, we developed several modifications options to improve the stability of donor oligo within the cell. This is the data that shows the HDR efficiency between the unmodified, phosphorothioated, or altered modified HDR donor oligos at multiple sites in human and mouse cells, which indicated in the blue, gray, and green color respectively. Under these conditions, HDR with the unmodified templates have less than 2% of the HDR rate. One level of improvement varies by site. The phosphorothioated modifications offer improvement, and the altered HDR donor oligos further enhance the improvement, ranging from 8 to 20% at these suboptimal sites. Now, I would like to switch gear a little bit to talk about the toolkits that are available to analyze the genome editing efficiency. The T7E1 assay recommended for people doing the knockout experiments, the PCR-based assay with primers flanking the edit sites. If editing has occurred, the PCR will result in the fragments of different lengths where the indels have occurred. Heating the amplicons generates the single-stranded fragments, which can then annulate to each other upon cooling. This will create the heteroduplexes, which can be cleaved by the T7E1 enzyme. And this cleavage creates the products of different lengths, which can be seen on a gel as different size bands. If only one band, that means no editing occurs, indicating the unsuccessful of the experiment. RAMseq is the most in-depth editing analysis tool. The amplicon sequencing around the on and off target edit sites is paired with a cloud-based analysis tool. Our RAMseq design tool will design a panel of PCR primers around the specified on and off-target sites within the genome. The RH primers have special RNA bases moiety and blocking groups to reduce the primer-dimer formations and applications of unintended sites. When the RNA base in the primer that binds with a complementary DNA base at the target site, the enzyme RNase H2 will cleave the end blocker from the primer end, allowing for amplifications of the DNA. This produces a much more specific, targeted PCR amplification. After cleaving those blocking groups and amplifying the first set of the amplicons, a second PCR is performed with the index primers to allow for a sequencer to read the data. The first step PCR adds these blue and orange groups which are complementary to the index primers. A second step PCR adds these index primers onto the amplicons to allow for a readout by a sequencer. And this method produces really quantified editing data and produces the nice graphics to demonstrate what types of editing is present. So, the RAMseq CRISPR analysis system is a new end-to-end workflow that we have developed to help the researchers quantify the effect of on and off-target editing using the NGS. This includes the developing RH primers for multiplex PCR using our RAMseq design tool, preparing the amplicon library using the corresponding master mix, and analyzing the sequencing data produced from an Illumina MySeq in an easy-to-use web platform. You can find more information on any of these components at the website that I shown in this slide. With the increased understanding to this system, researchers now have a better genetic and epigenetic control of cells and potentially to be used in a broad range of applications ranging from the generations of animal model, formations of the biomaterial and the biofuels, plant genome editing to increase the crop yield, gene surgery or drug development such as the gene and cell therapy. For more detailed information, you may visit our website with the links that show here, or you may contact us through the email. This concludes my presentations, and I will now hand over to Yunhee to lead the Q&A sessions.
Thank you, Edward, for that informative presentation. If you have a question and have not done so already, please type it into the Q&A box. Okay, there are a few questions for you. Here's the first one. What are the differences between Cas9 and Cas12a?
Thank you, Yunhee. So there are several differences between these two particular endonucleases. The Cas9 and Cas12a have different PAM recognition sites. The PAM sequence for Cas9 systems are either NGG or NCC, whereas the Cas12a is TTTV. At IDT, we established another variant of the Cas12a, which can recognize the PAM sequence with TTTN. So with this, it provides the researchers with the flexibility to target a broader regions of interest. Secondly, Cas9 requires the gRNAs at 100 nucleotide that consists of the cRNA and the tracer RNA, while Cas12a has a much shorter gRNAs at approximately 40 to 44 mers in total. At IDT, the gRNA of Cas12a consists of a spacer sequence at 21 to 24 mer together with the optimized conserved region. Additionally, the Cas12 system possesses the trans-DNA cleavage activity, resulting in a non-specific single-strand DNA cleavage.
Thank you, Edward. There is a second question here. What are the differences between wild-type Cas9 and high-fi Cas9?
Yep. The hi-fi Cas9 is actually known as the high-fidelity Cas9 nuclease V3, and it offers the reduced off-target effects offered the wild-type Cas9, greatly reducing the risk of off-target cutting events. For applications that are sensitive to off-target events, combining the high-fidelity Cas9 with gRNA is highly recommended.
Thank you for the answer. There is another question. In which systems have the HDR donor blocks been tested?
Yep, through our internal testing, our HDR blocks have successfully been used in mouse embryo microinjections, zebrafish, C. elegans, immortalized cell lines, iPSC, and the primary T cells. In addition to that, our altered donor blocks consist of the modification that help to mitigate the risk of blunt integration event. In next webinar, I will be providing a presentation about our HDR donor block with detailed information.
Thank you, Edward. All right, there is all the time we have for questions. I want to thank you, all of you, for attending today's presentation. I also would like to thank Edward for his informative presentation and quiz session. As a reminder, we'll email you a link to a recording of this webinar, as well as future webinars as they are scheduled. Thank you again for attending. We wish you the best of success in your research.